Nó nhỏ thì ơi,
Cannot hear.
nó hơi nhỏ.
Hello.
Rồi được rồi.
Okay, great. Check. Thank you. Oh. We have just few people to 20. So we'll copy the file to the computer. Great. So you will do great in home. Anyone?
Yes.
Okay, so we take about 10 minutes already. Just say hello. I know it's some of you is a kind of prepare for the thesis. Thesis is depends, maybe it's next or in next month. So few people is kind of busy. So I hope you doing well and your condition and health is great. Okay, today let me copy the file to computer. This is so our presentation today kind of short, I think, because we discuss quite a basic thing and many things you already know. So do you have anything to have any question from the last okay. Do you have any question from the last presentation for about the mutation and the way of the detection? So the way of detection, maybe somebody in the molecular diagnostic, we are really focused on already in many methods. And also for the COVID-19, so the infectious disease detection company really they fighting a lot and try to making a new method for new kit or new method for faster determine the infection of the virus for any country you need it. Vietnam also a success at some level. Okay, but the topic today kind of different matter with that. So we kind of focus on the genetics related disease in human. The detection maybe is later, so we will discuss more better. So the topic today, chapter 4 in the book I sent to you. So the question is, you already read the book, right? Some kind of chapter with the book, so you should have the written there. And my presentation also focus on the book. Just extract data from the book and make the here. If you have no kind of you want to deeper discussion, we can focus on the book and discussion with me or with your friend in class, but by internet chatting, not kind of face-to-face to chat. Okay, thank you. The topic today is autosomal dominant and recessive inheritance. So you still remember what is autosomal, right? So this is we have the 46 chromosome, and we have the 44, about 22 for each set of the chromosome set. So we have the 22 normal chromosome and 1 sex chromosome. So autosomal is from the number chromosome number 1 to number 22, a dominant and a recessive. So we also invert today. So I want to outline a little bit about medical genetics again. So the main target of our subject is a human disease, right? Human disease. Like I also mentioned about, we can apply to another species disease because of the, is human is the most important. Human is the most important. And a lot of money and a lot of the resources we spend on human research. But we can use that kind of information to apply to another species to improve our economy, our agriculture, and other things. So not only focus on human only, we use this kind of thing we can apply for another species for another specific purpose. So at first, I think this slide is made by my thinking. It's not based on any kind of book or making detract from really order from the book, just freely in my mind when I make the slide. So first we need to understand about the disease. So understand about the disease is not simple, because you have a book, you have internet, or you have something. But thinking about, for a long time ago, the scientists or the first one, maybe 100 or 200 years ago, the scientists and explorer, the first one, they don't have that kind of a thing. They have no book, they have no internet, and they have no any information about the disease. They can find themselves by logic or also the investigation. Very less assist from the technology. So understand, but now the situation is very good for you and also for me to understand about the disease, because a lot of diseases already have information, but still it's more than that. A lot of disease we don't have information yet, even though we know about the gene information, but we don't know which gene relate with which disease, and some kind of the monogenic or multiple genic disease. So that is the true thing. So the first is we find out about the etiology of disease. I mean the cause of disease by what? So if we have infectious disease, so this is mainly is because of the environment, right? Because the virus can, because of the we kind of have the interaction with some infected people. So that people transmit some virus to us and we infect us. And due to the genetic, like I mentioned in first presentation, due to the genetic of the people and also the condition of our health system, immune system, so we also have the relationship between the environment factor and also the genetic factor. And so what we have to find out is that disease because of the environment or about the genetic, because we know is right now, this is admitted information, a lot of disease related with genetic, including the infectious disease. And another is if you will point out that's disease because of genetics. So you have to answer, because we have a whole genome, it's very big entity, right? 3 billion base pair, and which part of our genome responds for the disease. So we have to identify which gene is disease. And first step is not simply which gene. First step is scientists identify which chromosome and which part of chromosome relate with the disease. So that's the information we can find out more clear in the chapter number 8. So identify the causative disease marker. And that's marker we use for diagnostic later. So which gene? So I mark it in here is which gene? That's a single gene or multiple gene. So that disease by using the effect, the outcome, disease outcome because of the single gene or multiple gene. And this is very hard part of the trying to understand about the disease. You know, is thinking about this new disease, and you don't know is it's because of genetic, and because of the environment is have no factor, not have the big impact to the disease outcome. So if you think it's because of gene, but which gene? So you're thinking, so you need to do some kind of experiment. So maybe compare genome between two populations, like this population, one population is suffer with the disease, and one population with the healthy phenotype. So compare with genome and identify some part of the some kind of what kind of reason of genome different between two population, and then you jump down in that genome position, and then find out more information about the gene. And luckily is we had the Human Genome Project, and another relay project is already solved the problem with understanding the function of the gene. So many genes we already know the function, and based on the relationship between gene function and the disease, it's the disease physiology. So we can make the link between the gene and the disease. And next thing, you can using some molecular cell biology technology to confirm your hypothesis, right? You need to confirm. Without confirmation, nobody accepts that kind of thing is true. And from that, okay. And next thing I kind of noted in here is the transmission pattern. So for example, the infectious disease by the virus or bacteria, we have to touch the source of the disease, the bacteria or microbe, right? We have to interact with that people have the virus to get that virus infect to our body. So the transmission. But in the genetic transmission, we're talking about from generation to the next generation. I mean, it's from my body to my son or my daughter. So how the pattern of transmission, that is also very important. Using that kind of, using this, if you know it's the transmission, you can do a lot of things. Like you can predict the risk of the next generation have disease or not, or how many percent, and the recurrence of that, the risk. So that's kind of term we can focus to answer or discuss deeper in the later slide of this presentation. And that is I'm talking about we do the task up there, try to understand about the disease, like the course of disease and the pattern of transmission of disease. Focus on the genetic related disease. So you have any kind of thing to want to clarify about the disease? You have any idea to got more information about the disease with this thing? Okay, you can post a question now or later just by type your question here or your idea. You can discuss, no problem. And the next thing, when you got the gene information and you know is about the mutation or the sets of the mutation, you can organize or design experiment for diagnostic, right? That is, we try to answer that people have the mutation and then they develop the disease or not. But you have to remember that one people have the mutation, but they may not have the symptom or any kind of expression of the disease outcome. Okay, we will discuss this in this presentation. And for diagnostic, maybe I just consider few people have the disease or not. Okay, but I kind of had a question about our population. So our population is a prevalent population. Like, what is the meaning of this word? So I concern, for example, I had one disease like thalassemia in Vietnam, or at least in Ho Chi Minh City. So I wonder that because the thalassemia is maybe had a very small effect or very big effect for the body. So I had a question: is in Ho Chi Minh City, how many people suffer with this one? So in the phenotype, or some declare some symptom, we can see some people express the disease, but for some people is a heterozygote. For some people in heterozygote, they don't express any kind of symptom of disease, right? So that is those people had a meaning or not? They also had a meaning because of they are carrier. They have that mutation in their gene, right? And they can transmit that gene to the next generation. So if you cannot control, all of the Ho Chi Minh City people will have that kind of mutation, and the society will be suffer by the disease by next generation if you have no strategy to control that kind of disease. So you have a strategy to control the disease, or you can use that kind of information to predict. Like my son or my daughter have a boyfriend or a girlfriend, so I can design whether or not I need to check before marriage, check the genetic first before marriage, because I don't want my grandson or have the disease in maybe this serious situation. And if you got already about the population in the prevalence of the mutation or the disease, you can estimate for the recurrent risk, right? Recurrent risk. So like mating estimation, or for example, you know, is that kind of disease or recurrent risk, you can estimate using the transmission pattern. This disease is a recessive or dominant. They are located on autosome or the sex chromosome, and based on that, you can do the service like counseling. So you can make the service and tell some married people around what you need to do in the next step. You need to do some PGD for checking your baby when he's or she was the embryo or not, or do some specific medication method for therapeutic or treatment. And that's also I mentioned here for Q or SS people. So a lot of diseases we cannot fix yet, because they are natively by gene already. We cannot change the gene for all of the cells in your body, right? You cannot fix that. But somehow in the development of the medical system now is very increased. So we can, some disease we can cure, and some disease we cannot fix, but we can access for the life better, or somehow make the lifespan of that suffer people is more easier for longer timeline. And for the higher and bigger picture of the disease management, and we have, maybe we can suggest some strategy for control the disease. Somehow you can make some kind of suggest and make some big project for testing people with the disease and makes diagnosis before they get baby. Then that is also useful for the society, right? I have some plan already. Okay, we have some question. Okay. Yeah, we right. We have the kind of pathology of disease. Jung asked about, we talk about the pathology disease development. Yeah, I accept that. So this is the development from gene to any kind of biochemical pathway, and we can make the disease more clear. That is obviously need for the disease understanding, from gene mutation to the phenotype. Thank you. Great. Okay. Next one is this is more focused on this chapter. So we just talking about a single gene disease. So we know that we have the I mentioned only the genetic disease, not the by environment disease something. So we have the multiple gene disease, and this is we will discuss in the next chapter. Maybe it's next week, or I can submit some video. You can we can discuss that. And mainly focus on this chapter is we'll talk about a single gene disease, or we call it monogenic disease, single gene disease. So disease by gene, but by single gene only, not by several genes. But remember, with one gene also is very big, right? Some gene is some hundreds kilobase, is very big gene. So some mutation is, we have a lot of mutation in that gene. So that's quite complex also. So talking about a single gene disease, so we have to mention about the OMIM database. OMIM database is a open medallion in man. So this database is start from 1960 by Dr. Victor Mikusic. And before that, just few kind of information he can gather. But later on, the number of the data is built up, and with the burn of the technology in molecular cell biotechnology, we explore more and more information about the gene and also the cell's activity related with the disease. So the data is now very quite a lot, but it's not complete yet. It's not complete yet. So this is the database. Yesterday I updated from yesterday. No, a few days ago. Already today 30, right? So in here is some statistics. This database right now is hosted by Johns Hopkins Medicine School in the USA. But also the database is really quite closely and shared by some big organization in the human human medical system or some biotechnology center in the USA. So like the NCBI or Ministry of Health of the USA also, they host data and they share data. And they also have another kind of database, and it track this data and in another data. So in here we have about this number of genes, more than 25,000 genes really. And we have the autosomal and X-linked chromosome and Y-linked chromosome disease. And also is a quite high number in here is that we have the gene related with the mitochondria gene. We also have some disease related with this one. So in here is a lot of genes. We just know information about some gene. Maybe in here we have the line here, very most described in the gene and phenotype combined already. So we have 35 very clear information here. And with the molecular basic know about just 5,000 genes. And some is we don't know about that information gene. As we have summarized in here. Because of the X-linked chromosome, also X chromosome quite big, we have a lot of genes related in this chromosome in here, and the Y chromosome becomes small. And many kind of gene is not have involved the disease in Y chromosome. We have to find here. So that is provide you quite a lot of information. Not a lot, but just an overview information in OMIM system. So I I think OMIM is already trying to commercialize that data because of the some company. I checked with some company working in a healthcare system related with the DNA variation. They also buy this database for the use for diagnosis for the customer service. I try to explore more about the OMIM system database, so you can use like this one. You can use this by your computer. So they have the search box. You just put your gene name and also the phenotype or kind of the disease name in here, and they can search for you all the information they have about the gene information, about the disease related with the gene, or if you put the phenotype or the disease, and then they give you a lot of genes related with that disease. And it's very good for the scientists and very good for doing the research related with this one. And what if I want to do it for animal? I also can work in this one because of the, is gene organization, no, sorry. The physiology and also the gene between people and animal is kind of quite similar. So we can use this one for another application. That's true. Okay. So at first I explored this one, gene description. So from this project of Human Genome Project, we kind of know about the sequence of the human genome sequence, but annotation, that's what we call this annotation. So annotation. Oh. Annotation. So annotation is trying to characterize the sequence, like try to understand that DNA code is do what work in the cells. That's it. So that gene is coding for what kind of protein, and that is we have what kind of exome or intron or something. I think it's a lot of you is already know this kind of term, right? In bioinformatics course. So from the Human Genome Project or another kind of project related with the annotation of the human genome. So this OMIM system just extract that information from another database, and they put also this one. So we have a gene name, we have what kind of protein, and relay with the localization of the protein in there. And you have here cloning expression, mapping of the gene, where is the chromosome number, and gene structure and gene function. and also they know for the animal model in here. So because of before doing the experiment on humans, so they need to do experiment on animal first. We cannot do experiment on humans. Seriously. So in here is I'm checking the second thing about the gene and phenotype combined. So there's more information, a lot of information related with the phenotype combined with gene, and they viewed by immune system. So in here they have this one information like allelic variant. So also including another feature of the entry. And if you go to the most, I think the most of the valuable data in the immune system is the pattern of inheritance. So we'll focus on this one today. So pattern of inheritance is very important, and I showed to you that is that kind of disease, that kind of phenotype, or make kind of the recessive or the dominant, and from that you can predict for the next generation more correctly, because the people is already do experiment or investigate very deep about this disease. Okay, so in here in the pattern of inheritance, in I checked one kind of the gene. You see here, inheritance pattern is that they say that very simple word that indicate that we need that kind of gene related with disease is related with the autosomal dominant disorder. Autosomal, not X chromosome, and dominant is not recessive, right? So using that information is very valuable for making their diagnosis, or at least estimate for the risk to the next generation. No question? So the thing is, I'm also interested about this database, and also in my lab, some student also they doing some kind of research related with the bioinformatics, try to use DNA variation to check to estimate the disease outcome for people. People is not expressed yet the disease, but we can predict for the future, because of some disease is they kind of late onset. I mean, it's from the young stage they don't express the disease, but in the future they will express the disease, because a lot of diseases are age-dependent development. So that thing is that kind of service right now is a lot, and I want to push some kind of and choice the game. I want to choice the game. So a few students in my lab is trying to do that, and he also in the class right now. And because of that, so I need a database. I need a database. So without a database, I cannot do that work, right? Because of I cannot do in my hand do a lot of research related with it. I need a database. So I need this information: autosomal dominant disorder, or recessive, or sex related disorder. So that help a lot. So I write email to the immune director or some staff, and they provide me home data. Very nice service, I think. But that data kind of we cannot. I have to use my email, like our university email, because I'm a teacher. So they get me because I want to do some research. Now, if you have the Gmail or Yahoo Mail or something, you cannot get it, because of they don't want to sell it, or if they sell it, just sell to a big company, I think. So I downloaded it for several months ago, and I think in several months some new mutation already submit and update to the database, and my database I got from the last several months is not quite new in now. But the settlement is not much, I think it's also quite new. Okay, in this chapter, we'll focus on single gene disease on autosome, and the sex chromosome or mitochondria we can discuss in the next slide. And we will discuss about a pattern of inheritance that is very important for prediction, or at least estimate the outcome of the disease in the specific people, or how to, or also very meaning to the family, right? And we also touch into some factor affect the inheritance. Because inheritance, you see sometimes it's a dominant or recessive. That is not very strictly correct. I mean, sometimes it's a dominant, sometimes it's a recessive in some case. So it's not correct. So we find out some factor, and like some, you know, Mendel, he is making some experiment, and a lot of people is including me, we believe in his the scientific basic of the experiment right now. We understand that, but a lot of people is not accepted because of they feel in their observation a lot of things is not correct. So they kind of fighting with the theory from Mendel. So, but in the book and in here, we've discussed about a factor affect the inheritance. That's maybe some observation is not strictly follow the old observation from the Mendel, like the recessive or the dominant. And we also discussed about some risks of the transmitting from this generation to the next generation, and some kind of real circumstance scenario in our society related with will affect the inheritance pattern, and also I mean some kind of event in a human society relate with inheritance. Relate with the frequencies of the disease, alleles frequencies in to prediction to disease. So any question for this part? Hello?
No.
Mm-hmm. Okay, so I go to the next thing. So we turn to some basic concept about the genetic. So we can discuss a lot about the rule of the Mendel. Because of we're talking about single gene, we didn't touch with the multiple gene yet, right? So we can the Mendel theory is quite correct in this case. So we talk about the contribution of Mendel in our course. So first, the most valuable work of Mendel is he made the three kind of genetic rules in for like he the father of genetics. So he made three rules, and when that time is nobody believed him. So no, I don't know if somebody is actually he his project is not knows a lot. So maybe it's 100 years, right? Many years later, people is noted about his experiment. So so first principle, first rule is he's talking about dominant and recessive inheritance. So in here is one kind of factor, one kind of the trait can dominant and kind of overreact compared with another phenotype. In here is we have the red and the cross with red with the white color, and now is we have the all red in 100%. That is the red is the dominant, and the white color is recessive. So dominant and recessive is quite famous when you were in high school, you know it already. So in the human phenotype expression, we also have the same thing. We have also the same thing. Same with the plant. And oh wait. So the second rule is a rule about separation. So remember is when the time of Mendel is he don't have any kind of knowledge about the gene, chromosome, or meiosis or something. Don't have it. Very long time ago. So but he is a kind of genius. He talking about genetic, the kind of factor making the phenotype is occur in pair. That is amazing, correct. Now we know is we have the two pair of the sets of the chromosome set. So any kind of gene we have the two version, one from mom and one from dad. So we have in pair. So that is I think is amazing locally. Logically suggestion. So this one is state that so any kind of so in pair and each kind of element of the genetic element will go to another gamete, and they can form its ladder because of become the zygote. So before Mendel hypothesis is accepted, so a lot of people is accepted blending hereditary because they said that the body will transfer a lot of things to the egg. The body will transfer a lot of things to the egg, and the egg is mixed with the sperm and transferred to the embryo. They in the Mendel we have the theory that is the theory of the chromosome. The gene on chromosome right now we know each chromosome in each pair will go separately. They not mix together and transfer something not clear to the embryo. And the third thing, third rule is the principle of independent absorption. So the genetic this phenotype will be transferred to next generation by the independently with another genetic factor. That's because of right now we know that the gene is that they localize the gene is localized maybe in different chromosome, and if they localize in the same chromosome, and if the physical location of two genes is close, they maybe go by their genetic link and go together to the next generation. But I had one question. I also had this question before. So how about the one set of the chromosome here? Through the generation to the generation, they can go in the one set, this whole set, to go to the next whole set. The set in here, the member in one set, they not change, or they keep, or they change, or they keep same member in that and go to the next generation. So somebody can answer that question. Hello? You understand the question?
Not really. Can you repeat it?
Okay. So, is in our chromosome, in our cells, we have a two set of the chromosome, right? One set is from mom and one set from dad, right? So that is question is that two set of chromosome. For example, I have in my germ cells, germ-like stem cells. So I have a two sets, right? And that sets will be go to from my father, and that set is from my father will go directly to my son, or in the germline stem cell they will mix together, and then they make a new set. Of course, the set is also including the number 1 to the number 23. They will mix it to make the new set, or that kind of set will be completely not changed and move to my son and my grandson later. Or not.
They mix.
They will mix.
They will mix.
Yeah,
Yeah.
and kind of reset it again.
Okay, correct. So because of that, the number of the sperm is unlimited, right? A lot of the we have a lot of number of the different sperm. Okay, great. So another one is also very basic. So we have a concept of genotype. So genotype is a combined between the the concept of phenotype. The phenotype is a combined between the genotype and environment. This we accept already. So this slide just provide some kind of more clear relate with the disease. Like the different genotype, we have the same phenotype. For example, in the cystic fibrosis, is kind of popular in Europe. So we have the AA genotype and big A and big A genotype and big A and small A genotype. They not express the disease, right? They same phenotype, even they are a different genotype. And another one is the same genotype but with different phenotype. For example, the mutation on the ketonuria, so PKU disease. So maybe last time also mentioned about this disease. One student in our class, or she doing some sequencing related with this disease. I remember I in the committee for her proposal defense. So she tried to identify the mutation related with this disease. So so the baby with this disease will have kind of defect in the enzyme related with the phenylalanine. I mean, it's one amino acid, right? Phenylalanine. So transfer to tyrosine. But the enzyme in this baby is kind of have a mutation and they're not expressed. So the phenylalanine, because of they cannot transfer to another form, so the phenylalanine is quite accumulate in your cells, and they make some toxic effect. So with the environment is low and normal diet of phenylalanine. For example, in the high amounts of meat or the animal or animal originate food. So we have a lot of phenylalanine. So it's low diet of phenylalanine. So we have the baby safer. And because of the phenylalanine in that food is kind of low. And if normal, so the toxic effect will be expressed. And that baby will be has a problem with the nerve system, and the IQ will be decreased. So that's the concept of phenotype. And next is we have to familiar with the pedigree structure of the human. So pedigree structure quite similar a human to if you are using on animal also the same thing. But the human is more clear and a more okay. This is a circle is a normal female circle and have no any dark inside. So in the square we have a normal male, and in this case we have a sex not specified with this symbol, and the symbol will indicate mating. And this is a traditional family, like this first generation and this second generation with the 3 tribes. So, and this one is the partner of this male is they have no choice analysis. And double bar indicator, they kind of close mating, close mating between the sister and the brother in the same family or kind of closing together. And this is twin, and not identical twin, and this is identical twin. And this is try to minimize some symbol in the pedigree, multiple individual for each family, same sex. And this is a more, we focus more circle and a square with the dark inside. So this is related with the disease phenotype expression. And if it's not clear, but they have a dark in the middle, so this is a carrier. That means they are heterozygous and they not express the disease outcome. And this is that. And SB is a stillbirth. I mean, it's we have some baby, and no, it's not baby yet. So the fetus is die inside of the mom belly. So about 20 week gestation, we noted this here. Okay, I finished for the last thing. About the proband, we had the first people we have diagnosed with the disease phenotype. And we also consider the relatedness of the family. Maybe the first degree of the relatedness in the family, maybe the sister and a brother very closely. And the second, maybe the cousin together. And the third is you can see that maybe the grandma and grandson. And close or far relationship in the family. Okay. Now we will go in the main dish of the chapter. We'll talk about autosomal dominant inheritance. So have a look at this pedigree. Have a look at this pedigree. So we have the white color, female and male, and this is not expressed disease, and this is expressed disease. So we're talking about autosomal dominant inheritance, and I want you to look at this one and check it. So what is characteristics of autosomal dominant inheritance? Can you write some kind of something you notice about this pedigree? Random assortment. Random assortment. So assortment like Mendel, right? But the Mendel is to say that is we have the independent assortment, so it's random. We have no any kind of close marriage in here, close relate marriage in here, right? They both they quite different. We have a 3 generation from the grandma in here, and we have the parent, and we have the granddaughter and grandson. You can see something from here. You know, it's from checking the pedigree, you can refer to. You can check that that is what that disease is quite of what kind of pattern of disease. The pattern quite simple. If the pedigree is viewed quite correct, this is the perfect for the making the conclusion about the pattern of the inheritance pattern of the disease. So at first, you can see that is dark, square or circle. You can see that men or the women, both, they have the disease, right? Men or women, they have the disease. And we have that means that something, you can say that they maybe not relate with the sex chromosome. They maybe not relate with the sex chromosome, right? Because of if you have the just man, oh, sorry, this one too hard. In the pedigree, so in the pedigree, if we just have the man, got disease, that means this quite relate with the Y chromosome, right? But in here, men and women, they also express the disease. If you have the pedigree with all the men in the family express the disease, that means also relate with the X chromosome, right? Because of a women, we have XX, and maybe another X, it will cover the mutation of the error gene in the remain X chromosome. But in the Y, in the man, we have the XY, and there are the mutation in the X chromosome, and they have no other copy of within the Y chromosome to confess with that kind of side effect of that chromosome. So in here you can see that both men and women in this pedigree, they have disease. So they maybe not relate with X or Y chromosome. So they should maybe tend to be they belong to the autosomal chromosome, right? Okay. And also it's important is we see that 1, 2, 3, 4 women and also 4 men infect with the disease. So 50 and 50 is look like they are randomly on men and women. So maybe it's not related with sex chromosome. Okay. And another is you can see the first generation, second generation, and third generation. Any generation have the people with the disease. That means the expression of disease is kind of strong. And they come from maybe the homozygote of the dominant or heterozygote, they also express disease, maybe. And that is make you thinking about the dominant of the disease, right? So that's quite a kind of sign for you to see that is inheritance pattern of this pedigree disease is autosomal dominant, not recessive. Because the recessive may be the father or parents, they don't have expressed disease because they are carrier. Maybe they are, we can check it later, but maybe they are here. They have A, maybe A and A. So in this case, they have no kind of express any kind of disease, but their offspring, they will produce this one, small a and small a. And some in the generation, some kind of some generation we will lack of the disease. But in here you can see that all generation they have a disease. That means dominant, not the recessive. Okay. So any question about this one? Not yet. So just have discussion. If you have any question, just focus. We can discuss. I'm not sure I can answer the question, but we can try. So this is their characteristics of autosomal dominant inheritance. So like we said, that two sex exhibit the traits in the equal ratio, right? So it's not related with the sex chromosome linked. And then no skipping of generation. So if you have it. So your mom or your dad will have that kind of disease. So no skipping. Skipping is something high already, and they show up in next generation. And father to son transmission of the disease gene is observed from the father to son. Check it, from father to son in here, father to son. So father to son is not necessary to conclude of the dominant autosomal dominant inheritance, but just rule out another pattern. And another pattern, like in next chapter, we can see is father to daughter only, father to daughter only, like X chromosome relate. And the final one is maybe also is not required in this pattern. That is half of child will affect. Half of child will affect. Okay. Half of child will affect. Why in this situation like this? Because of the I also show it's the most common case of mating. Because this is dominant. So in heterozygote, they will express their dominant. Now is a big A, uppercase A is the dominant gene, the disease gene, right? So normally is the case of the, because of the, you know, it's this dominant. So the most scenario of the making the offspring with the disease expression is made from big A and small a. That is and the parent with the uneffect, that means small a and small a. And because of, if we have the big A and big A, maybe that case is not happen because of this is dominant gene, dominant mutation. And dominant mutation, if you will make a big A and big A together, maybe the effect's very strong and they kill that people already. And the most scenario is we have the normal parent, and mate with this normal parent, and mate with the effect parent, and using that you can see this. You have this ratio: 50% will affect with the disease, and 50% will save with the disease. Or some case, some case you have the effect parent and effect parent will make it. So in here is now we also no. Now is we have the, because of big A and big A's maybe die already. So we have the heterozygote about 2:3 ratio, right? Because of the parent kind of they see each other kind of in a society wide they kind of close together. Maybe they get married for example. Very rare case. There is a lot of case is the dominant inheritance will make a very big effect to the body. So maybe the reproduction they not system maybe not work very well. So they don't make a new generation. That is also one aspect. This thing. Sorry. And this is one kind of disease. It's Autosomal polydactyly. So we have the number 6 finger or tail. So I can see some case in here in my eyes for a long time ago. And last year also had a dream with one people with he had just three finger. He had 3 finger. I think he anything normal, but he kind of short, and he had just 3 finger nature, but not by cutting. Okay, So that's the kind of characteristics of the autosomal dominant inheritance. So you have any question? Question? No? Okay. So we talk about the recurrent risk. Recurrent risk is the probability, the ratio of the children will have effect. We have not the children yet, so we can predict about the risk of the children can have the disease or not. Because of, you know, is when if I have the disease, I also very worried for my baby. He or she will have the disease with same with me or not. So I don't want that kind of effect, right? So I want to know the recurrent risk, and before that I can design I want to have a baby or not, or at least I have to, if I have a condition, good condition, I can use some kind of technique or medical technique to rescue the situation. Like I can do some kind of diagnosis for my and my wife embryo. Yes, the baby embryo, for taking one cells and checking by some kind of technique, and that embryo is safe, and they go to the next step for making a baby. So in here we have autosomal dominant. So normally is a one half of the child will be each child will be for example, my first child, so he had the 50% got the disease, because this is dominant, and we also is discussing here how children will affect in this scenario. So if I affect and my wife also affect, I mean the ratio will change, but that thing is maybe very rare to happen. So 50%. So this case is the most. And this can also maybe sometimes happen in our society, because they are kind of blow each other in the situation and then they get married. But this one is maybe this big A and big A will be got the die, and we have the two, maybe 2 per 3. This one. And how about this one? This one will not happen, I guess. Okay. Why don't we have a little break, maybe just 5 or 7 minutes before we go next thing, relate with the recessive inheritance. Okay. Break. Question, Nam?
No.
Thank you. It's feel lonely in here, really. So, do you have any question related with the report question? I make it from last week. So maybe some research already shows the relationship between thalassemia and malaria. So we just search it and explore more about this kind of stuff. Make it short. Okay, what we know about the autosomal dominant inheritance. So we can move to the autosomal recessive inheritance. This is just quite a short describe it in the phenotype in the pedigree. You can see it too. So in here we have also 3 generation pedigree. So we check that out. So the this pedigree I downloaded from internet is we have the black color in here, like affects people. I mean, it's a disease have phenotype out, especially. I mean, it's some phenotype we cannot see is in the observative trait, like the number of finger, like the polydactyly before. Some kind of phenotype we have to check using the biochemical checking, like this one, PKU, like amounts of the phenylalanine accumulate in the blood, or some specific tissue, like kidney or something. I mean, if the phenotype have to express in the observative phenotype, or clinical, we have to test with a specific test, like the enzyme amounts or some kind of relate with some it's not expression outside. So here we have a carrier, like kind of brown color and dot, a lot of dot in the center like this. Like a carrier, that means is they have the mutation, but they not express the disease. Yeah. But thinking about this kind of thing is we don't know about a carrier situation. When we investigate a family pedigree related with the disease, we just know about their white color, clear, or the dark. Because we didn't use any kind of diagnostic, I mean, it's a gene diagnostic for checking the carrier or not. So this is maybe not true, or at least this one is a very deep investigation. All of the member in the family or in the pedigree need to be checked by gene, by checking the mutation. And this is quite clear. And this is the end stage of the research already. So beginning, you don't have that information. We can check by clinically the phenotype expressed, and the carrier situation, we don't know. So in here you can see that men or women, they both have the expressed disease. That means related with autosomal recessive. Remember the X chromosome. X chromosome also men or women, they also express the disease. Like the recessive disease in the X chromosome, men or women, they both express. But all the men will be expressed in the X chromosome. We can discuss deeper in the next week. But in here you can see that we have 2 people in the one generation, but they express, and a lot of people is not expressed. But in here we have the kind of curious compare with the dominant inheritance. That means in dominant, from if I have the disease, I mean my mom or dad should be having disease, right? So in here, these people have disease, but their parents have no disease. That means it's different with this case. This one is 2 sex inhibiting exhibit the trait. But we have a skipping generation of the expressed disease, right? So I mean, this generation don't have disease, express only this generation. That's it shows that's related with the recessive. Recessive is in this generation. This is a high hidden by the dominant. The dominant is normal, and the recessive hide themselves in these people, and they express will become when they become the form of a homozygous of the recessive disease mutation, right? So quite easy to get this information. So from this one, you can suggest or diagnose, I mean, the gene diagnose for anybody in the family. So you can grow this kind of picture more clear and grow in your project if you want to do this kind of research. So characteristics of autosomal dominant inheritance is they may be not expressed in earlier generation. You will see this one, and we have a lot of cases not follow this kind of rule. Not follow this kind of rule. Maybe we can understood. No. We cannot distinguish between the. Okay, sorry. This one is recessive. Sometimes we cannot distinguish between recessive and another pattern, another error we will see later. So maybe we have the skip expressed in some earlier generation. Male and female are affected in an equal proportion, right? This is not related with sex chromosome. And finally, maybe the consanguinity. We will explore this term in the end of the chapter. This is kind of serious thing related with the human society. That is consanguinity is related with the close relates mating between the mating between the close related people, like brother and sister. So that happens more often in this pedigree. So also because of this thing that because we are human, not like the animal. So the human is have kind of some specific characteristics. Some kind of cases are most common, and some kind of cases are maybe not actual happen. So in the recessive inheritance, the most case is a carrier mating with the carrier. And you also have the question related with maybe the carrier, maybe big A and small a, mated with the small a and the small a. But that case, small a and the small a maybe is expressed very serious phenotype outcome. So they may not join to the married case. They cannot marry because of the weak kind of is that people is physiology, biology is the reproduction system will not work very well. And another one is the society related with marriage, that people is not really visible, right? Maybe they're very short, and maybe they express some kind of serious thing in expression outside, and nobody wants to marry with them. That's the crucial. And you can see that I put some one example in here. So the disease I'm talking about albinism related with tyrosinase negative. So that gene, this gene is quite a heavy defect, has a mutation, and that gene is not work. So they cannot. This enzyme is not present in that people, while present in very low amount, and they cannot transfer tyrosine to melanin. You know, is melanin is kind of an accessory to make the pigment in the skin, right? So the people is a kind of had a white color. Even in here, we had African women. He had a white color in her skin, and also because the melanin is also the one important component in the fiber in the eyes. So the people who suffer with this kind of mutation will have the phenotype of the eyes. The eyes muscle is kind of is not stable, and the eyes will be moving a lot. They're moving, or sometimes is they become a strabismus. That means what the black part is not localized in the right axis. They may be bent a little bit in the left side or another the wrong axis in the eyes. So this kind of here you can see that the color and this one colors. So the eye is not symmetric. Okay. So you can see that in here we have the fourth generation pedigree, and you can see first 3 generations don't have expressed any kind of disease like this, and suddenly we have the one close related people mating together. We have 4 children, and we have 2 child express the disease. So you see that this is kind of relate with a lot of kind of resemble with the characteristics of autosomal recessive inheritance. But here we have the men and women got disease and close mating, and we have ancestor people don't express disease. And the dot in here, the carrier, of course, we have to test in, if we have to investigate, we have to type in genotype, DNA checking for those member in the pedigree. Okay. So recurrent risk of this one is about 25%, right? 25%. Different with the dominant, we have the most common case is small a, and big A and small a. We have a 50%. And this one is we have the carrier mating with carrier, and the offspring about 25%. Okay, but in here we have 50%, right? Because of what? Because of the then you have to remember in the last week we already calculated already. So the probability of each child will always independently with the next child and independently with the next child. So in here we have the if 2 parent is heterozygote and 55% we have reproduce a homozygote of the disease form, small a and small a, and 25%. So and the next child will also 25%. In this case, we had a 50%. But if this couple make more children, this will be go more close with the 25%. Okay. And in this case, we have the this one is about carrier and may with the affect people. In this, some kind of disease affect people is still is mating. And in here we have the recurrent risk. In this case, about 50%, and 50% maybe is assembled with autosomal dominant. But if you more if you more investigate about the disease, is more close with the more clear about the pattern of the inheritance. And how about the 2 people with the disease mating together? In that scenario, maybe kind of low, because of the remember in first report from you. So the mutation rate is kind of low, right? Kind of low in the society, and normally they not met each other to mating together. So I have one question related with how about the prevalence between autosomal dominant and recessive? So how about which one is more compared with the other? So dominant or recessive, which one is stronger in the actual society? Question. So the prevalent is the amounts of those people in the current situation. So do you think is dominant is a more dominant disease is compared with the recessive is more, or recessive is more compared with the dominant? You get the question? Recessive. And why?
You mean prevalence mean being common?
Yes.
Yeah. So I think the recessive one is more common because the dominant individuals will be die earlier, and if the recessive alleles can be transferred
to next generation.
Yeah.
Okay, that's correct. Good. So that is also quite similar with your first report, right? So the natural selection, and also in human we have the society selection also. So is asking somebody in here, you accept them to get married with somebody is have the defect in the gene, maybe you don't accept it. That's true. And another one is this is just a warning. So remember those information is maybe not available for you because of there's some kind of disease or kind of phenotype you don't have information about, right? You don't know is that kind of you have to identify that is a dominant or recessive in the sex chromosome or autosomal dominant. If you have information, simple, it's quite quite simpler, more simple, right? But if you don't have any information, what gene, the pattern of the inheritance, that's more complex, and sometimes, and I think it's almost time, it's hard to identify that that's the case. And we will go more deeper in the actual when you try to identify the pattern of the inheritance. So here is I take some mention about the dominant and recessive. Sometimes it's not correct, and you have to careful when using that kind of thing. And also, when you were a researcher, or if you want to investigate some kind of new disease, you know, I want to investigate a lot of disease in our country because of like in the book. You can see a lot of the disease, but relate with the European people only, not Asia. But Asia, maybe in China or Japan, a rich country, but our country, we don't have that much. You know, it's a lot of people with the black in his face in Vietnam, but I don't know if we have the last kind of investigation, some research, some study about the disease in our population that we don't have that one. That's what is also fascinating, I think. So talking about a dominant and recessive, you have to strictly careful when using. So in here, I state from the book, I state that the dominant and recessive is not clear due to the disease. Why for that? So you know, you see from the last presentation, we had a mutation, and based on the mutation, and based on the location of the mutation, I mean, maybe the mutation in the gene body, and you got the interrupt gene, and that gene is not totally lost 50% or 100% amounts of the protein, or if the protein and if the mutation occur in the promoter, so the number amounts of protein will be decreasing and variable in the amounts of protein. So I mean, it's related with dose of the protein to the cells, right? Maybe 20%, 100%, or 200% sometimes. And also is another more important about the roles of protein in the cell activity. Some protein maybe not necessary. Some protein, I mean, no, a lot of protein is kind of, some protein is joined to very specific. I mean, it's in the end of the biochemical pathway. In the end, maybe it's not very important. But if that's kind of protein, there is the enzyme, but in the center of the biochemistry of the cells. So I mean, the role of protein is increased because they involve a lot of the pathway. So you have a problem, right? So the importance is of the protein is different to the another protein. So I have some example in here. So this girl is have an achondroplasia. So the mutation, that's the autosomal dominant. But this girl is even they see her body kind of not mature because she have the short stature of the limb, and also she kind of short in her height, but she have a nearly as a normal life, maybe just a few years less than normal case. So I mean is in this is in the kind of dominant heterozygote, she not express too much about the lifetime. She can living healthy, right? And in heterozygote of some recessive disease, remember the heterozygotes of the recessive, so we have no any kind of side effect, right? We say we have normal body, but remember in some, because of the you have the mutation, you have the heterozygote, you have the bad mutation in your gene, your genotype. So the amounts of that protein in your body also decrease, and in some case maybe it's dangerous, right? So we have no kind of absolute dominant and absolute recessive. They have some effect in some hands. It's for clarifying. And how can we check that kind of situation? And if you, for checking, so if you increase number of sample for checking and increase number of the case, like dominant is the always effect and the recessive is an effect. But if the side effect is kind of vary related with recessive. So any question about this term? So no question? Okay. Another one is they also very complex about gene mutation. So we have some kind of calcium mutation related with dominant or recessive. So for example, one example I take from the book is growth hormone deficiency. So you know, it's our cells, our body needs growth hormone. So without a growth hormone, we our state is very short because no development, normal development. So this relate with this gene, GH1 gene. So GH1 gene is we want to check the size of the body between you see different species, or different phenotype between short or tall people and big and small animal. With one of them is we check this one first. And one recessive mutation is destroy one copy and remain one. So people is still normal. They just a little bit short. I mean, in here, even in one gene, but we have a different type of the mutation, different type of mutation. But due to the mutation and due to the effect of the mutation, we can turn that kind of we can say that gene is recessive or dominant related with the phenotype. In here is recessive is if we have one copy, we also save, no problem. And in case of this is recessive, they express when the small a and small a match each other. When its dominant mutation happen, in last presentation I also show it, this dominant negative mutation. I mean, the allele number 2, we make the protein, and this protein is not contribute to the main function of the growth hormone in here, but they will produce a new protein, and this protein will be interfere the activity of the original protein and make the work of this protein become quite a near to zero, and that is expressed to the phenotype. So this mutation is become the dominant mutation. I mean, it's big A and small a, they also express to the phenotype. Okay. So I also had a question like, you may not have info of the toxicity mutation because of in the immune system, they only list some kind of mutation. They already do some research, but we still have a lot of mutation because of not all the people in the world have the gene checking. So still have a lot of in specific population. Maybe in Vietnam we have some kind of mutation related with that gene or specific gene. So this slide shows that the gene and the disease related with gene is dominant or recessive is also based on their specific mutation. It's not always the dominant or not always the recessive. It's complicated, right? So maybe in the OMIM database, they also show that kind of the situation because this is a book already. So in OMIM system, maybe that gene related with the dominant, or that gene, that mutation related with the recessive. Another mutation in same gene is dominant. That is also normal. Okay, so we kind of, this one is a constant of the dominant and recessive. You just know about the recessive inheritance and dominant inheritance in the autosomal. But the life is not beautiful that much because of that's just in the book. But actually, it's quite very difficult to identify the pedigree with the clear information about the disease like that. We will go some factor affect expression of disease kind of gene. How to express this one? The recessive and the dominant not always correct, and hard to identify that expression because of some factor like this. And the first factor I'm not listing here. First factor is and also is that is actual doing the research. That is the phenotyping. So here. Phenotyping. So in the research, phenotyping is one of the hard part, not just design primer, not PCR, not sequencing. That is simple. Somehow you can success it. But phenotyping is more important. And you know, is what is the most important in genetic? That is sample. Without sample, Without human with the disease or healthy, you cannot do anything, right? Without a mutation or without information about the phenotype, you cannot make any conclusion about the disease. Phenotyping also very complicated. Right example is some kind of phenotype kind of simple to measure, like the height of people. You can just put that people to the wall and put the ruler, and you can get that phenotype. Simple, but some kind of phenotype you cannot identify more correct because of data now is continuously, and how you can identify which one is weak, which one is strong, which one is low, which one is high. It's quite hard to identify that kind of situation. And also, for example, some kind of bones fracture. I mean, the calcium in the bone also is hard to identify. You have to use very good machine to do that. And also, for example, you measure the mineral inside your bone. And first, today you measure the amounts of the machine is will generate this number in today, and tomorrow they will generate a different number, different values. So you see, phenotyping is the most complex task in the research related with this field. So somebody talked about if somebody see some nature paper, very big paper, it's simple. It's simple. These experiments are very simple, but they can go to Nature or the Lancet or New England Medicine because of their sampling and also phenotyping. They spend a lot of money for that, and best people in that project, they can go that high, and not BCR or something. Okay, so I go to the first factor related affecting the expression of this current gene. So first is a new mutation. New mutation is like the picture visualize that thing. So the parent don't have a mutation, don't have any kind of disease thing, but the baby have it. The baby have it. So that can make us quite confused with the autosomal recessive, right? Autosomal recessive also the parent don't have the phenotype, but the baby have it. They have a skip generation expression. So the phenotype not occur in the parent or ancestor. And one of the characteristics is kind of rare, right? Because of you see that the mutation is the event is rare event. It's not happen is spontaneously, and normally they cause the dominant disease. So if the mutation happen, so the disease will express, and we can recognize it. So for example, is most of the case of the short stative, the girl in the picture here. So it's related with the new mutation, and the disease rare will be increased in the next generation. Of course, because of the baby right now, they make from the sperm or the egg from mom, and that is the egg or sperm. They had a mutation, and the baby had all of the 100% cells of the baby, including the germline stem cells of this baby, also had a mutation, and he can transfer that kind of mutation to next generation. So the disease rate will be increased in next generation. Because of the last generation, we don't have this disease. And next thing is germline mosaicism. So you can have a question like germline mosaicism is what is different between the what the difference between germline mosaicism to get versus the new mutation. So you have 2 or more offspring present the mutation, right? The thing is not like the mutation, because of the mutation is a rare event. So like this father have this sperm mutation, and this father again make another mutation for another sperm. That's not maybe not happen. So that's related with the in the germ cells of the father, in the germ cell population of the father, he already have the mutation, and they can make maybe portion of the sperm have this mutation. Some is normal and some is abnormal, and that's also increase the the frequencies of the mutation in the next generation. So this because of embryonic development of the parent. So remember, it's different with the new mutation. The father is normal, totally normal, but some mutation happen in some sperm of his germ cells. So, but in here, in the germline mosaicism, is his sperm cells, his germline stem cell has a problem from when he was an embryo already. Okay, and this also no history of disease in pedigree. So his father also don't have it. And so, okay. And another factor related with the expression of disease causing gene. So that factor is make your conclusion related with the recessive or dominant is maybe not correct. It's also related with reduced penetrate. Reduced penetrate is the term we talk about the person who have the disease phenotype, the disease genotype, the gene, but they do not develop the disease phenotype. That's also happen. That may be related with interaction between the genotype and the environment. Maybe the environment very good, so he don't express the disease. And also maybe relate with the interaction between the gene in his body. For example, people had a bad gene, but another good gene, different locus. So also can compensate for that kind of bad gene. They can cover for the bad gene, so that people is not expressed to the disease. In here, so we have the disease retinoblastoma. So that is autodominant disease, autosome and dominant, and make the cancer in your eyes, But in here, in a big investigation in the big population, the scientists identify about 10% of the carrier, obligate carrier. Obligate carrier is the term we're talking about. The people have his next generation had the disease, and his previous generation also have disease. So he must have the disease genotype, because the next generation and previous generation also have the disease. So he must have it in here. So we have these people. We have the big A and small a. This is autodominant, so he should have expressed the disease. But in here, he don't have it. And another people will have expressed disease. So this is, we say 10% is don't have the disease. So the penetration of this disease is about 90%. It's not always 100% of people with the disease genotype will express the disease. Okay, question? Yes, maybe the environment. Okay. Environment or some nutrition or some kind of the medical system in that place is good. You know, it's a lot of things is not clear in this field. We try to answer the question, but a lot of stuff is not clear yet. And another one is related with age-dependent penetrate. So it's kind of simple. A lot of disease in the young stage, we don't have expressive disease, but in the older and later periods, we express the disease and higher penetrate. So for example, the Huntington disease here, so they express, we express, I take this one from internet, it's some kind of the Rowing is not mainly focused on this, but this some kind of disease in here, Huntington disease, they just expressed in high old people only. Another disease, Alzheimer's disease. Young people don't have disease, and many kind of the dominant cancer is only expressed in the late stage of the lifetime, like some kind of ovarian cancer or ovarian cancer or some breast cancer, family breast cancer. Just old people have it and young people don't have it. But a lot of cancer right now is younger and younger. So this kind of the phenomenon is make the harmful effect to the society because of the young people did not express the disease. Oh, sorry, a lot of wrong. The young people, they have the genotype. I mean, they are carrier. They bearing the genotype have the disease, but they don't know about that. They don't know. They don't know, so that they desire to get a baby. They desire to get a baby, and this because of this is dominant. A lot of things is dominant. So his baby will have that kind of disease, even though he don't want it, but he don't know about that because of young stage he get married and he get a baby. So that kind of thing will be increase the frequency in the population of the disease mutation. And because of the young the delay in the delay defend the age in the people had this mutation. So this one reduce the natural selection because of a lot of disease is expressed when we were born. That means it's a newborn defect. But in here, some diseases are not expressed in the newborn. So the nature is not remove this one out of our system. Because of that, so a lot of the quite a number of disease related with this one is late onset. And also is I also had thinking myself. That is when you make the pedigree, if you want to do some research in this one, so you make the pedigree, and if you have some baby in there, if you have baby, and you think it's and also you have to think about you don't have information about disease. So the age-dependent penetrate, you don't know about this diseases have this phenomenon or not. So you also include some baby in your phenotype, in your pedigree, and then your prediction may be not correct. Just warning about this case, right? So no question. Okay, so that is also one factor. Make your pedigree information and make your conclusion related with the disease inheritance pattern of prediction will be wrong. Another one is a variable expression, and that also is also simple. It's also back to the story about dose of the protein. So based on their mutation location, the characteristics of the mutation, the expression of the protein will be changed in various level. So the disease outcome also will occur in a different variable outcome expression. It's variable expression is kind of different with penetrate. Penetrate is we have yes or no have disease, and variable expression is we have different level. Maybe the very small effect to the body or very serious effect to the body. So one of the characteristics of this thing is related with a very big gene. For example, in here we have the NF1 gene, neurofibromatosis. A lot of gene is named by the disease they relate. See, NF1, so neurofibromatosis. So this is related with some tumor in the brain. So this many they contribute to the disease is related with the new mutation a lot. Because of this gene is very big, very big. So about 350 kb, very big. So you know it's become a random mutation. So the big gene is more suffer compared with small gene, right? Big gene is suffer easier compared to small gene. Like they jump to the big land is easier to jump to the small land. So a lot of mutation contribute to this disease, and their different level from small to the big effect to the body. And another one is also easy to image is a locus heterogeneity. That is, the disease can cause by mutation at different loci in different family. For example, you know, it's a biochemical pathway of anybody is kind of same. But a lot of some gene maybe contribute to the same, maybe the same, some different gene is contribute to the disease, to the pathway, to the biochemical pathway. And this family maybe have the mutation with one gene, but contribute to that pathway. Another family have another mutation with another gene relate to the same pathway. So in here, one example is here APKD disease, polycystic kidney disease. My kidney is really terrible like this thing. So have a lot of tumor in the kidney. And they code by the gene on the chromosome 16. This is PKD1 and chromosome 4 in the PKD2. I also have a question related with this one. Maybe this gene, sorry. First, I need to mention this. This is two gene related with encoding for two protein. They make the same function in the cell. They make for their they make the one channel in the cell membrane channel. So they change the ability of the go in and go out of the sodium in the cells, and it most effect to the kidney. So you see that different chromosome gene, but they have the same effect. But we may have a question, maybe this belong to the same gene, same family, gene family. Maybe that is two genes is a kind of globally. These two genes kind of globally have the relationship, maybe the gene duplication, and somehow in the evolution they jump to different chromosome. That may be also the common case, but I didn't check this one yet. If you want to check it, you can check is those locus heterogeneity is those genes make the disease, they really belong to the gene family or not. And you can have the answer for that. Another is of pleiotropy. That is one gene. It's reverse way with the locus heterogeneity. This one is many gene, different gene have the same effect. But the reverse way is one gene have the multiple effect. That means, like we mentioned before, so one gene, they in the center of the biochemical pathway in the cells. So the different in the gene, different mutation in the gene, maybe go to another pathway. This mutation of same gene go to another pathway effect. So one of them example is mafan syndrome here. So we have the mutation in this protein. It's the main component in the connective tissue, and you see that the lymph and the tissue of this boy kind of stretchable, and it kind of longer than normal, and quite very flexible so they are not strong enough to maintain the fixed body for the boy. This is also the second case when the thumb is not in the right position than normal. Because of this protein is very important, so they contribute for the eyes, the skeleton, and also for the blood transfer system. And that is normal happen in the gene-related disease because of you see is cystic fibrosis also the same thing because of the disease affect to many organs in the body, and many organ that means is many pathway. Okay, and we go to the end with discussion of some kind of phenomenon is common in the human society. That means is consanguinity in human population. That means is the close relate married. So it means the mating between the close in genetic people. And this is a map in the world map. The white color, that means they don't any kind of research at Vietnam in here, but I think Vietnam is still good because our rule is strictly allow get married between the same family people. But in here in Africa, we have a serious situation, and also in India also. In South India also becomes very serious situation. Several years ago, and I go to USA. I go to USA for the conference in Utah state. And in Utah, it's kind of in the middle of USA. So from beginning of Utah, some family come to Utah, some family only, and in there they have the religion allow the one man can get married with a lot of women. One man is like the heat of the very first population in Utah is he had his name is Brigham Young, I remember. So he had more than a lot of wives, and he had a lot of children. And he also is the head of the religion. So you think about his gene is transferred to the next generation, and this next generation can make together. And you see that what is expected this situation. Like the first report I question to you is we have the mutation. Nobody in our planet we have the perfect genome, right? We have no any perfect genome. So we have some defects, somehow we have some bad gene in our genome. And then, but we still normal because of luckily we have two sets of DNA in our genome. So the one normal set will cover for the bad set. So we have normal in almost a case. And sometimes is almost common is we have the recessive. They express. But most case we not express because of the recessive is high by the normal. So if the race of the clone relay marriage, they will expose that kind of the bad gene out of the hiding, right? So you can expect that the disease phenotype will be increased because of the bad gene is not high anymore. And this is some research, some study related with that. This is our consanguinity and relationship with the frequency of the recessive disease. And the dominant maybe they express and big A, and big A is maybe is become the very dangerous for the embryo, and maybe it's not make the baby. They're not become a full term and success. So the first effect will be increase the chance of mate with the mutant carrier, right? So if the brother and sister mating together, or at least in the same pedigree people mating together, so they increase the chance. Change to mate with the mutant carrier, right? Increase the prevalence of disease in the population also. For example, in last week, we already say that cystic fibrosis in Europe, just 25 people, we had 1 people is a carrier. Quite high, right? Quite high. So we have a 1 to 25 chance to match the carrier. I mean, if I a carrier, or if I normal, I also met the carrier is out of the 25 women, so I met 1 people with the carrier of disease. But I don't know if this one is a 25 or 50 here. Because of the I'm man, so I need to choose women, right? So this number it will be increased a little bit. So if the first causing that means is very close. So the second relationship, second degree in pedigree relationship. So we increase to 1 to A, 1A. So we increase a lot. So the chance to make the disease offspring will be increased. And how about the compare between the disease? Some is common disease, some is a rare disease. And if your friend or you see some case and they got some genetic disease, and if that disease is very rare, so that means that kind of the probabilities of the parents of that people, diseased people, is kind of they have a close relationship in gene, right? For example, in France, they do some research. So cystic fibrosis is about 1.4% in the offspring for the first causing mating. Very low, quite low. Because of the cystic fibrosis is kind of common in Europe. But compared with another disease, more rare disease is like cystinosis and chromatosia, like the increase the process. This means that's people disease. That people is they identify the case like this, and this is fraction of the case. They come from the parent have the close relationship, and this also showing the statistics about the relation people like this one is unrelated and more close in genetic. So the mortality is increased in the close relationship. Okay. So in here unrelated, so the mortality is just 8%, and this one increased to 40%. And they also increase the carrier in population, right? Okay, we go to the end of the lecture. And the next, we will learn about the sex chromosome related disease, and also with mitochondria, it's quite more complex to understand. So I will send my PPT first to you next week. Okay, any question related with the okay, any question? We kind of finished already. No question? 10 question? Explain the protein dose again. Yeah. So the theory, we have the dominant or recessive. Dominant is we will have the disease, right? We will have the disease. We will have the disease. And where is the slide like that? And in the case of the the case of the heterozygote, okay, the heterozygote, because the heterozygote is kind of quite of maybe the carrier or the most common case of the mutation can present in our population, heterozygote, big A and a small a. So in heterozygote, so in case of the dominant inheritance, dominant is that people is maybe have the fully disease. But the thing is fully diseased, or in case of the recessive is heterozygous, have no disease. But the barrier between two terms is not always correct. That's because of the protein dose, right? Because the protein dose, you say that we had a mutation in the gene, and the effect to the protein dose, or completely the effect to the protein dose. In here we have the in case of we have another case like the amount of protein is reduced, but in here is one alleles they completely no generate any kind of protein, and now is we have the half the protein amount, maybe 50%, but the 50% may be also enough for the cell can work, for the body can survive well and express no any kind of phenotype in the disease in here. But in case of the maybe more focus on the case of the heterozygote in the recessive. So recessive, we say that people have no suffer any kind of disease. But the protein dose right now, 50%, we just have 50% compared with the normal people have the 100%. So at least we say that people is not suffer anything about the disease, but it's maybe not correct. He or she will suffer about with the disease, but maybe the effect may be small because of the protein dose is not enough for her cells anymore. In several cases, if we change the environment, that protein dose is maybe more is more important to him. So its disease will be expressed is more dominant. So that is related with the protein dose, related with we cannot say that absolute recessive or absolute dominant. Okay. It kind of confused. But the protein dose is I just mentioned is we have no absolute dominant or recessive. Somehow is in some case we have a dominant, and some case we have a recessive. For example, in the this is we found already about sickle cell disease. Sickle cell disease. So sickle cell disease in the heterozygote, because sickle cell disease is a recessive disease, so only expressed where we have a small a and small a's, right? Normally we have the small a and small a's is very serious and they die very soon, maybe very short lifespan. But we have the heterozygote, big A and small a of the sickle cells thing. Then their living is also normal. But the problem is when he changed the environment from the lowland to the highland, I mean, it's the oxygen amount is kind of low. Maybe he go to the Himalaya or he go to the Everest climbing. And that's thing from the recessive will be changed to the dominant, because the oxygen right now is not enough, right? And the recessive phenotype and genotype will be changed to the dominant because of the he will die soon because it's not enough the oxygen for his works amount in the Everest. Okay, any question? Okay, if you have no any questions, so we stop here and see you in the next week. And I will try to produce some video because of be prepared for the midterm. But the thing is, the midterm and this year, this semester, we just count for about 25% of your score. So also we have to spare something for the final. Okay, have a good day, everyone. See you soon, I hope.
Bye, teacher.
And have a good time in your home. Thank you very much for today. To start it.